Now, the biotechnology, the application of microorganisms in biotechnology, such as genetics, engineering, and bioremediation. Or the application of microorganisms in technology. There's a difference between application of microorganisms to produce antibiotics or to produce, the citric acid or things like that in industrial biology. This is different. This is the microorganisms used in technology, or technologies used on the microorganisms, such as genetic engineering. This is pretty concise, and genetic engineering is a common theme of the biotechnology. In genetic engineering, we do what? We usually take the plasmid. Since plasmid is small in size, so it won't be overloaded, and we cut it and take the genetic material of some other organism which has the property that we desire to be, and that microorganisms. We combine their genetic material and another characteristics appear, in scientific words, another phenotype appear. Of course, the genotype is changed by us humans, and the phenotype appears by the microorganism. And that was the phenotype that was desired by us. For example, the bacteria didn't had the penicillin producing ability, but we needed that. So we just took the genetic code from that could produce the penicillin and took it into the placed into the other microorganisms. Now you will be asking, why do we have to take it from the other organisms? Why can't we use the other microorganism as it is? There are multiple reasons. Machinery is too difficult, or it needs a lot of nutrients, or its life cycle is very long, and things like that. So we select the microorganism that is very low cost, like it don't need nutrients that much, or it can live in harsh environments, or it doesn't need that much pH maintenance or things like that. And then we just have to put the genetic code.